Good evening, everyone. Welcome to today's webinar on HIV and the importance of using fourth-generation testing. We are thrilled to have all of you here today as we dive into one of the most critical advancements in HIV diagnostics. Fourth-generation testing has been around for a while, in fact, more than a decade now, but there are advancements that are happening on a day-to-day basis. And as the HIV epidemic continues to affect millions of people worldwide, and early detection remains one of the most effective strategies for controlling its spread. Over the years, significant improvements have been made in the accuracy and timing of HIV tests. One such advancement is the fourth-generation HIV test, which has transformed the way we approach HIV detection. We're very pleased to have with us today Longjam with us, and with him in this session, we will explore what makes the fourth-generation test different, its benefits, and why it's essential in diagnosing HIV earlier and more effectively. This webinar will also cover how this testing technology can detect both HIV antibodies and P24 antigens, allowing for earlier detection within the critical window period. We hope by the end of today's discussion, you will gain a deeper understanding of why embracing fourth-generation testing is crucial in preventing the transmission of HIV, as well as improving the quality of care for those affected. So without further ado, let's get started on this vital journey of learning. Over to you, Dr. Longjam. Namba.
Thank you, sir. So today we are going to have an overview of this HIV virus, why it has been regarded still as an HIV epidemic and it's a threat, and the use of fourth-generation tests for the diagnosis and screening, and above all for the prognosis stage of these HIV-infected individuals. So before going to the details about this HIV, I would like to summarize today's objectives of today's session. So we will be discussing about the timeline of the HIV. So herefore, it has been 40 years timeline of the HIV since from its discovery to the mankind, and overview of the HIV, how it looks, how it occurs as an infections, and the global epidemiology of this virus, and regarding some antiretroviral agents which has been established and creating a great impact on treatment, and the lab diagnosis, which we are mainly focusing in today's session. So before going details about this virus, I would like to highlight about that up to 2024, we have covered the journey of 40 years timeline from its discovery of this virus. So this was first in introductions and discussed in the year 1981, where Los Angeles hospitals has hospitalized 41 patients, where they consult with the Luc Montagnier, who was the pioneer of this virus, and he has the pioneer of discovering this virus to the mankind. So it was started in the 1981, but when in the year 1983, it was discovered that the lymphadenopathy associated virus, which was more problematic issue in the public health impact during that 1981, it was introduced in 1983. Similarly, in the year 1984, just 1 year after, in the United States, Robert Gallows, the scientist, he has discovered that how the pathogenesis mode of transmissions and how the pathogenesis of this virus related with that public health impact of that lymphadenopathy associated virus. But at that time, they termed it as, it was not finalized and specific that it was a unique different virus. So they were associated with the similar functions and the properties of this human T-cell lymphotropic virus. And from the name itself, we come to know that they are loving to the T-cells lymphocytes. So that's why they have named it as human T-cell lymphotropic virus. And if we see the timelines up to 2023, there are lots of landmarks has discovered and has given by the many stalwarts and the pioneers scientists who has done in the lab diagnosis, which was and the antiretroviral agents. Until now, we are in the era of these 21st centuries, where we are trying to end the AIDS by the 2030, and it was resolutions taken by the United Nations AIDS or the World Health Organizations that it is achievable if we keep our efforts strong. So this is the summary of the previous timeline, where I can show you that in the year 1981, a very rare disease, rare disease, which we call it as Kaposi sarcoma, and pneumonia caused by a pneumocystis carinii. At that timeline zone, this was considered as a fungus, but now it has been studied as a parasite. So now it has been taxonomically put under the parasites, and it has been named as pneumocystis cerevisiae. So it was creating a havoc in the Los Angeles, and mainly they were found in the homosexuals or the addicted to the IV drugs users. And then later on, it is named as the acute immunodeficiency syndrome, which where they are having the problematic issues with these 41 hospitalized patients. And in the year 1983, the stalwart pioneer scientist, Dr. Luc Montagnier, and his colleagues has isolated this virus, and they have named it as lymphadenopathy associated virus, because mainly the symptomatic infections of this virus were related with the persistent generalized lymphadenopathy. In the meanwhile, within 1 year, the United States, Robert Gallo and his colleagues reported these isolations of this similar retrovirus from the AIDS patients, and they call it as the human T-cell lymphotropic virus due to loving and tropisms of the T-cell lymphocytes. And in these timelines, we can see that in the year 1986, the Committee on the Virus Nomenclatures International has given or decided the generic name of this virus as human immunodeficiency virus, because this virus was related with the human immunodeficiency system. And later on in this 1986, another serotype of HIV, which we now known it as the HIV type 2, it was isolated from the West Africa. So within these 3 years, when in the discovery of the first HIV type 1, within these 3 years, this virus has generated another serotype by the genetic variations as known as the HIV type 2. And in this year 1986, our India has reported the first HIV case. And due to this public health impact, in the year 1992, the first National AIDS Control Organization, NACO, was constituted. And in the year 1996, with the stalwarts and the high committees, they have considered that in spite of giving a single treatment therapy, they will be possible to give a high active antiretroviral therapy, which becomes a combined therapy, and it's a new standard for the care or the treatment for the HIV. Now, coming to the 2017 to 2024, this NACO has India launches even year national strategic plan on HIV or AIDS, including the sexually transmitted infections. Now, before going to the in details, we want to highlight an overview of this virus, human immunodeficiency virus. As all of us know, as a general concept regarding this virus, that once it has been infected, the infected individuals has contracted this HIV virus for a lifelong. Then we also know that it causes havoc syndromes disease that is known as acquired immunodeficiency syndrome, commonly known as AIDS. But I would like to highlight that this AIDS is the final end stage of the clinical stages or the infections by this HIV virus. So we must not create a misconception that when we acquire the HIV virus, it will definitely not lead to AIDS, but this is the end last stage, which is due to the HIV-induced deficiencies in the immune systems. So in the timeline, we have discussed that the HIV was first isolated by the end of the 1983, and HIV type 2 is discovered in the year 1986. And these HIV2 are more prevalent in the West Africa, whereas the HIV type 1 are the most commonest HIV serotype, which are prevalent in all over the world. Now, if you want to further know about this, I would like to have a taxonomic classifications of this virus. Here we can see that belongs to the family Retroviridae. Retro means a back, going back. So this virus has mechanisms of an reverse transcriptase, which just violates the central dogma of life. All of us know that the central dogma of life says that from the DNA, the RNA will be synthesized, and from the RNA, the protein will be synthesized. But this retroviridae has got a special enzyme which is known as reverse transcriptase enzyme. Due to the presence of it will violate the central dogma of life. In spite of synthesis of RNA from DNA, these reverse transcripted enzymes will enable this virus to transform or synthesis the DNA from the RNA. That's why they belong to the Retroviridae family. Under this Retroviridae family, there are two subfamily orders, which we consider it as Orthoretrovirinae and Spumaretrovirinae. This Orthoretrovirinae, under this Orthoretrovirinae, the human pathogenic human T-cell lymphotropic virus, HIV type 1 and type 2 belong to this genera lentivirus. Why this lentivirus? Because they do look like other lens pulse. And the disease associated with HIV type 1 and HIV type 2 are the end stage AIDS. And whereas this human lymphotropic virus type 1 and type 2, previously this HIV-1 was considered as human T-cell lymphotropic virus type 3. But in the year 1986, it has been considered designated with a generic name HIV. Now, as all of must know that the virus, the infectious virus particles outside the target cells, we term it as virion. So the structures of this virion of this HIV are spherical, as we can see this scanning electron micrograph of this virus, where we can see that they are somewhat spherical in nature. And the diameter of this virus is around about 90 to 120 nanometer. And here we can see in this scanning electron micrograph of this HIV virus that there inside there is a cylindrical core which we can see with this scanning electron micrograph. This cylindrical core consists of a nucleic acid with some protein known as capsid. So collectively it is known as nucleoprotein, and these are the symmetry of these nucleocapsid proteins are icosahedral. And under this, the virus is composed of ribose nucleic acids, that RNA, in spite of DNA. So all of us know that the virus will have presents only one single nucleic acid. So it may be either DNA or RNA. So this HIV virus belongs to the RNA virus, where RNA only constitutes as a nucleic acid. So diagrammatically, if we represents this spherical HIV virus, so diagrammatically we can see over here that it has got an envelope. So they are belong to the envelope RNA virus, and these envelopes are lipid in nature. Under this, on this envelope, we can see a spike proteins, and these proteins are glycosylated. That is combinations of the proteins with carbohydrate moiety, and they are known as glycoproteins. So these glycoproteins has two subparts. One, just like the mushroom, it has known the glycoprotein GP120, which we consider commonly as a surface proteins, and the attached to it, it is a transmembrane protein, which has got the glycoprotein 41. So, and inside this HIV virus, if we open up the lipid envelope, we can see that they are surrounded by matrix proteins. So this matrix protein surrounded the cylindrical core, which is of icosahedral symmetry, and this nucleocapsid core has composed of the protein capsomeres, and collectively it is known as capsids proteins, and they are covering or they are protecting the two identical linear RNA strands, where we can see, and they are associated with small proteins like integrase proteins and protease proteins. Now, if we see the compositions of different biomolecules, compositions of this virus, we can see that they are mainly made up of proteins, about 60%, and lipid is around about 35%, and carbohydrate is very less as compared to the lipid and proteins. It is of 3%, and the nucleic acid is 2%. And yes, lipids natures are mainly having a protective covering that is known as envelope. So this is an envelope RNA virus, and there are also envelope RNA viruses, but this HIV virus belongs to the envelope RNA virus. Now, coming to the genomic structures, that is, we are going to go in details about the RNA nucleic acids of this HIV virus, and that is the HIV genome. And we already know from the diagrammatic representations that they are composed of two identical subunits of single-stranded RNA. So they are diploid, and these RNA nucleic acids are not circular, which we mainly found in the bacterial chromosomes, but they are linear, and they are positive strands. There are two types of strands of messenger RNA. One is positive sense and the another one is negative sense. And this one is the nature of positive sense. And this genome, if we compare with the other retroviruses, which we have discussed in the previous slides, like human T-cell lymphotropic virus 1, 2, and the Sp'2024 virus, they are more complex than that other retroviruses. Why we consider it as a more complex, that if we can see here from here the diagrammatic representations where we highlighted the differences genome representation of HIV-1, HIV-2, and the other human T-cell lymphotropic virus, which we commonly have comparisons with the other retroviruses. Here, what we can see is that this MA stands for the matrix protein. So when we discuss about the a diagrammatic representation of the HIV virus, we come to know that there were the matrix, which was covering the cylindrical core nucleocapsids. That matrix proteins has been synthesized or produced from this segment of nucleotides, and this is known as MA, matrix protein. CA stands for the capsid proteins, which we have discussed in the previous slides, and the NC stands for the nucleocapsids. And this PR stands for the genomic strands or the nucleotide sequences, which are responsible for the protease enzymes or proteins, and these RT stands for the reverse transcriptase and IN stands for the integrase. And there are other regulatory genes along which we have designated with the pink color. So these are the regulatory genes, or it is also known as accessory genes, which will helpful in the regulations of the virus replications in the infected cells. And the green color is these are the envelope genes only, which are only meant for the proteins or the other glycosylated proteins which are present on the envelope. So here we can see that the blue color considers of these matrix or capsids or nucleocapsids, and this red color stands for this involving or revolving around the nucleic acids, and the pink color are the accessory or regulatory genes, and the green colors are the envelope genes. Here, I would like to highlight that the main difference between this HIV type serotype 1 and HIV type 2 genotype, the main difference is this accessory genes, which is BPU, and the another one is the BPX. So the BPX is present in the HIV-2, and the BPU is present in the HIV type 1, whereas the BPR and BIF are present in both the HIV type 1 and type 2. And if we see in the other retroviruses, here we are taking the example of the human T lymphocyte virus type 1. What we can see here is that these VIF, VPR, VPX, which are one of the accessory genes, including the NEF, they are totally absent in these other retroviruses. Here we can see very clearly, and there is the another that transcription activator translator proteins. They are also absent. In spite of this, that in the other retroviruses there is TEX, and in spite of regulatory virus proteins REV, in the other retroviruses it is REX. So these are the differences between the other retroviruses and the HIV virus in the genomic level. So here we can also see that collectively the retroviruses, the gene order in the nucleic acid is arranged as 5' GAG gene, which is designated with the blue color, and then PRO, which stands for the protease gene, and the POL, which stands for the polymerase genes, and ENV, which stands for the envelope genes. So they are arranged in this way in all the retroviruses. Now, coming to the HIV type 1 genome, we would consider to highlight under 3 categories so that we learn it very easily. So the 3 headings are the group-specific antigens, sorry for the spelling, so this must be specific, group-specific antigens, and type or subgroup-specific antigens, and other regulatory genes. So in these group-specific antigens, we come to know from the name itself that they are specific for the group of that HIV, means they are both present in the HIV type 1 and HIV type 2. So what are they? Number one is the GAT, which we have just now studied in the blue color, but here in this picture, now it is in the green color. So the GAT co-proteins are responsible for the productions of these protein 15, protein 17, and protein 24. So these 15, 17, 24 are nothing but a wake, because we weigh the proteins in the kilodalton. So when the protein weight is 15 kilodalton, then we designated the protein as P15, and if it is 17, then P17, and 24 as this, the weight of this protein is 24 kilodalton. Now coming to the pol genes, which are responsible for the reverse transcripted enzymes or polymerase enzyme, which is the specific and most important enzyme that specifies that they belong to the Retroviridae family. And these pol genes, moreover, it also synthesizes the protease genes, and it is considered as the P16 as well as the integrase protein, which is weight of P31. Now, coming to the type or subgroup specific antigens. So this type of subgroup specific antigens are present and synthesized by the envelope gene, and this envelope gene will synthesize the glycoprotein 160. If it is disintegrated or breakdown, it will have the 2 counterparts. One is in the outer membrane part, which we have discussed as the surface proteins, and it is GP120, and attached to the GP120 surface protein, there is another transmembrane protein, GP41, and the other regulatory genes like that, REV, then VIF, this is your viral infectivity factor, and NEF, negativity factor. So this factor NEF will regulate the how far these viral proteins has to be synthesized. If there is excess, then it will generate activate NEF, and then it will down regulate the productions of this virus, VPR and VPU. Now, if all those discussions are simply in this diagram, where we can see the length of the nucleotide. So roundabout, it is about 9,700 kilobase nucleotide, one sequence. So this is about only one RNA single strand, where we can see that the GAT will have the P53, and it will again disintegrate into P18, P24, and P15. This P24 is very important. Later on, we will discuss in the lab diagnosis about this P24, and this P24 are signifying or synthesized in as the nucleocapsid proteins. And for these polymerase which are surrounding around the nucleic acids, it's a P160, and this reverse transcriptase is P66. And remember one thing, for every author, by every scientist, the weight of these proteins varies. So some will be saying P66 and someone will be saying P55. Likewise, the GP160 here we are considering as a GP40, but in most of the textbook or articles or journal, there you will find as a GP41. So it is just a number where it is just indicating the weight of the protein. Now, under the pathogenesis, I think, and I'm 100% sure that all of us know that the mode of transmission of this HIV. So one by one, we know that most of the HIV are spread through sexual transmissions, and that sexual transmission is not all the behavior of the sexual transmitter. It totally depend upon the behavioral of the sexual transmission. So if it is unprotected sexual contact with the infected HIV positive person, so there is highly chances of HIV transmissions. And blood or the blood products and the organ transplantations, there also we could get the HIV virus. And the third one is the most common in the healthcare setup. And in the healthcare setup, it may be due to the injury of by the needle stick or the contaminated needles by the healthcare workers, and via the vertical transmissions. So vertical transmissions, all of us know that it is transmitted from the mother to child. It may be during the pregnancy or during the delivery or after the birth of the child. So it may be during transplantation route or perinatal or postnatal transmissions. But we should consider it also that most of has a misconception that they might be transmitted by the casual contact, kissing, or through insect bite. So let me clear you that they are not transmitted by the casual contact if the skin is in contact skin. If there is an ulcerations of the skin, yes, there is a risk of transmissions of this HIV. But if it is the casual contact through this direct skin contact, so there is no evidence of these transmissions, or there is no evidence via kissing, or there is no evidence via the biting of the insects, because the bite of the insects doesn't transmitted it. Rather than biting, they only suck the blood. So they do not transmitted this virus by the biting of the insects. Now, coming to the uh, relative risk factors and types of the risk factors. So we have presented you in a tabular form. In this tabular form, what we can see here is that the second column signifies the relative risk percentage of the transmission per exposure. So here in this column, what we can see is that the transmissions via the blood or the blood product transfusion is very high. So in the per export, there is a high chances of more than 90% if we compare with the sexual intercourse. But if we analyze the statistical analysis of the transmissions occurred, here what we can see is that the most highly number transmission is by the sexual intercourse, but the risk factors is just a very few numbers, that is 0.1 to 1. But due to the ignorance of the sexual partners or the multiple sexual partner without any unprotected, these transmissions has got increase in number, that is 75%. And in India, it is around about 88.9. And from mother to child, we can see that relative risk factor is lesser from the blood transfusion. But if we see that the relative risk percentage factors for this blood transfusion or blood products transfusions, they are very high in number, even though, but the percentage of contributions to transmission in the World Incidence Report or the Indian Incidence Report, it is very less. Why so? Because now there is more stringent vigilance of the screening of the blood donors or the organ donors. So that's why the percentage of total transmission is very less. And that's that involves the importance of the diagnostic test. So if we increase the diagnostic test, there is less chances of total transmissions, when it will contribute to the less prevalence of this HIV virus. Now, how it generates the infections. First of all, we have to consider these first steps, that is the HIV. Here we can see one HIV virus is infecting the host cells, and this host cell is none other than the lymphocytes or the T cells, which we particularly specifically term it as T cells. And this HIV virus has got the surface proteins, what we term it as glycoprotein 120. Remember 120 and GP41, that was the transmembrane protein. The surface protein GP120, it loves the CD4 T cell surface receptors present on most of the T cell lymphocytes. And with this, with this, they will bind or they will attach with the HIV virus. So after this attachment of this virus to the target cells, there will be a helper, and that helper is of two molecules, either CC chemokine receptor type 5 or CXC chemokine receptor type 4. When this HIV virus has the capability of binding to the T cell receptor, this CXC chemokine receptor type 4 acts as a helper molecules to further attach and fuse this virus to the target cells. But remember one thing, these CD4 T cell receptors, the surface receptors, are also present in the macrophages or monocytes or other lymphocytes. So if binding on that molecules, then the CC chemokine receptor type 5 will help as a co-helper. So there was a misconception that it only infect the T helper cells, but it also can attach or infect the other cells like monocytes or lymphocytes or even the dendritic cells, which is considered as an antigen presenting cells, which are mainly present on the skin. So they are also have a domain of attachment or the binding by this HIV virus, mainly via the GP120. Now, when there is an attachment, then there will be the next step, and the next step is the fusions of this whole envelope, the fusion of this whole envelope to the target cell. So when there is the fusions, this is activated by the helper of these transmembrane proteins, that was the GP40 or GP41, which was present as a spike proteins on the envelope. So when there will be the fusions over these target cells, then there will be the release of the cylindrical core, that is nucleocapsids core proteins, and when there will be direct translocation to the nucleus. So here we can see from this diagram, the third diagram steps are that the viral DNA is formed by the reverse transcription. Before integrating into the human host cell chromosome, before integrating, they will go to the reverse transcriptions. So in this reverse transcriptions, the main helping role of this reverse transcripted enzyme plays an important role. So due to the help of this reverse transcriptase enzyme, the viral RNA will develop into a viral DNA. So this viral DNA, which was developed from this viral RNA, this is known as the proviral DNA. So this proviral DNA will to the infected human host cell chromosomes, and when this integrated, so it is enhanced by the integrase enzyme, which we have discussed in the previous slide. So when it is integrated into the target human chromosome cells, then later on, since this is the provirus DNA, so with there will be the generations of the viral proteins, replications, translations, transcriptions, and all these subsequently replications, transcription, and translations of the viral proteins will be there. And then there will be the assembly of those synthesized viral proteins, and then there will be the synthesis of the daughter cells from daughter cells, and then the last one will be your budding of these daughter cells by lytic cycles, and it will lysis the target host cells via budding from them, and it acquires the lipid envelope. That the lipid which was enveloping this viral RNA, basically it is acquired from the human host cells, and all of us know that the human host cells has got the cell membrane of phospholipid by nature. Now, coming to the clinical manifestations. So, according to World Health Organizations, it has been classified into 4 clinical stages, whereas due to the CDC, classified into the 9 stages, but most of the healthcare workers, they are considered these 4 clinical stages given by the World Health Organizations, and this is a tabular form which we can correlate with the number of the CD40 helper cell count per microliter. Here what we can see is that the these HIV infections, first, if there will be the HIV infection, here there will be no compromise of the CD4 helper cells, so there will be no decrease of the CD4 helper cells, and in which we can see it as a asymptomatic, but there will be persistent generalized lymphadenopathy in this HIV clinical stage 1. Later on, if it is untreated, left untreated, then it will go on to the clinical stage 2, where we can see that the CD4 T helper cell count is decreasing up to 499. Here we can see that there will be a moderate unexplained weight loss of the infected patients, and there will be recurrent respiratory tract infections, and there will be the infections of the herpes zoster leading to herpes or shingles if it is a reactivations, angular cheilitis, recurrent oral ulcerations, papulopruritic eruptions, and the fungal infections. So, and again, going to the advanced, there will be the chronic infections that is known as the third clinical stages of these HIV infections, where we can see that it is further decrease the CD4 helper T cells ranging from 200 to 349. Here in these manifestations, we can see an unexplained severe weight loss. Previously, it was more moderate weight loss, and here we see the unexplained severe weight loss and the unexplained chronic diarrhea for more than 1 month. And then there will be unexplained persistent fever, oral candidiasis, oral hairy leukoplakia, and there will be the pulmonary tuberculosis. Mainly it encounter mostly in the Indian scenario, where the tuberculosis is epidemic in every region of India, and there will be severe bacterial infections by the other bacteria, and there will be unexplained anemia, neutropenia, and or including the chronic thrombocytopenia. Now, the last end stage or the full-blown acquired immunodeficiency syndrome, where the WHO has kept into 4 clinical stages of the HIV infections, in which we can see that the T helper cell CT counts has down-regulated less than 200 cell count per microliter. And here we can see the infections. These opportunistic infections are those infections by the microbes, and all of us know that the microorganisms are present everywhere, so in nature, so they are ubiquitous, and mainly they are trying to invade the human system. So when the T helper cells, the immune system has been lesser than 200, then it is viable to achieve their target, so there will be most of the opportunistic infections in this stage. Now, due to the advance in science and technologies, and most of the pioneers, scientists, they have lots of advance in the antiretroviral drugs. These antiretroviral drugs, in order to study, if we have discussed the pathogenesis role, where we can encounter that most of these drugs have been targeting the entry inhibitors here. Sorry for the spelling. So the entry inhibitors means the attachment of the GP120 and the fusion of the GP40 or the 41. So if it is targeting that stage, then the drugs has been established and known as NFU-VTD and Maraviroc. And here what you can see is that there is an asterisk mark. So these asterisk marks are those antiretroviral drugs which are available in the NACO program, mainly in India. Now, for the integrase inhibitors, means those that integrase enzyme which likely to integrate the pro-viral DNA which was developed from the viral RNA. So they have to integrate in the target human chromosome. So in order to inhibit that steps, raltegravir, dolutegravir have been available in the market. And there is a nucleoside reverse transcriptase inhibitor. So reverse transcriptase, and let me tell you, nucleoside are those these
Yeah.
sequences of these adenine, guanine, uracils, and all without the triphosphate. So nucleoside reverse transcriptase inhibitors. So these are the avacavir, zidovudine, or AZT, and the diadenosines, jalcitabine, and lamivudine. Zidovudine was the first antiretroviral agent which was introduced or discovered by the mankind, and it was pioneered by the United States. Now, coming to the another antiretroviral drugs groups, which is your non-nucleoside inhibitors, and they are the nevirapine, etravirine, and efavirenz. And coming to the nucleotide reverse transcriptase inhibitors, these are the tenofovir disoproxyl fumarate, which is available in the NACO program India, in the other developed countries, tenofovir alafenamide are also available. And for the protease inhibitors, so these protease are the enzymes which are helpful in degrading the GP. So when the enzymes, the corresponding enzyme GAG genes will be responsible for the GP160. So it has to be degraded into GP120 for the surface protein and GP41 for the transmembrane protein. So that will be inhibited by these antiretroviral drugs, shakinavir, ritonavir, indinavir, lopinavir, atazanavir, and darunavir. So these are the asterisk drugs are available in the NACO program India. Now, there is a slogan or taken up by the United Nations AIDS or the World Health Organization for the ending the AIDS epidemic as a public health threat by 2030. So why it has been strategized and have been taken up by the stakeholders, because HIV is not an easy virus to defeat. And more than 600,000 people are still dying every year from this virus. Why? Because most of us don't know they have the HIV. So this creates the importance of screening of the HIV. And most of these HIV infected people, since they don't know they have the HIV, they are not on treatment, or they started treatment too late. So these are the most problematic issues which we are encountering, and it is contributing this HIV as a public health issue. So why still the HIV epidemic is not over? Because it has been started in the year 1981, and now we are in the 21st century. Most of it we are still fearing, and the stigma and the ignorance. So this is a photograph taken and most famous in the year 1990. Here we can see an infected HIV that his name is David Covey, an HIV or AIDS activist nearby to his death, surrounded by his brother and his sister-in-law and niece, and they are the inhabitants of Ohio, US, and it was taken in 1990. Copyright, the photographer was Therese Frere. Now, if we see the HIV data and statistics, and it is updated in the July 2024, it has been available in the World Health Organization's website. So here what we can see is that here total people living with HIV, it is encountering at 39.9 million. Within this bracket is the minimum range and a max upper higher range. And here what we can see is that this 39.9 million is contributed by the adult, that is above 15 years of age, 38.6. If we segregate into the gender wise, here what we can see is that the women is contributing more, and if we compare with the men, and the children, which is less than 15 years, it is contributing 1.4 million. So contributing all this 1.4 million and 38.6 million altogether worldwide, there is 39.9 million people living with HIV. So these people living with HIV will have the higher chances of acquiring HIV as total 1.3 million. And if we want to segregate into the adult and the children or the gender wise, this is the statistical data report. And after this, the statistical analysis, the people dying from HIV related causes is around about 630,000. So still there is an epidemic or stigma or ignorance of this HIV. Now, this is a graphical representations data from the World Health Organization updated in July 2024. Here, what we can see is that the World Health Organization has divided the whole world into the 6 regions: the African, Americas, Southeast Asia, Europe, Eastern Mediterranean, and Western Pacific regions. So if I would like, as in being an Indian, we would like to consider as a Southeast Asian, where we can see that 4 million people are living with the HIV. And globally, it is 39.9 million. And here what we can see is that the Eastern Mediterranean are lesser in numbers, 530,000. And let me inform you that the Oman has reported that in the year 2022, they have no reports of the any HIV cases till now. And here, this is your summary of the Global and World Health Organization regional HIV epidemic, where we can see that globally, it is number of people acquiring HIV is the 1.3 million. And for the Southeast Asia regions, mainly why I'm focusing here, because India, as being an Indian, we are still in this. So here it is contributing 1.2 million, and this number of people dying from HIV related causes is 83,000. So it's a huge number. Now, this is the whisker box chart, which are targeting by the World Health Organization's regions in the year 2023. So they have a target, and that target is known as 95, 90, and 86. This is the target, 95, 90, and 86. So what is this 95, what is this 90, and what is this 86? We are going to understand by seeing this. So this blue color is the target for the 95, and this green color is target for this 90, and the saffron color is for the targeting of the 86. So this column is for the globally, and these are the region wise. So here Southeast Asian regions are here. If we analyze the Southeast Asian regions, 95 target is that the percentage of people who know their status. So the World Health Organizations give a target that 95% of the people globally should know the HIV status of their own. So here in the Southeast Asian regions, we are still standing in the 78 in spite of 95%. And there's 90% percentage of people on antiretroviral treatment. So this will come when there will be the target of this, when the people know the status of the HIV. So this is of the 90%. And when they started treatment, this CD4 helper T cells viral load suppressions will has to be 80%. So this saffron color will talk about the viral load. So the viral load target by the World Health Organizations by 2030 is 80%, and the antiretroviral therapy treatment will be your 90%, and the knowing the HIV status will be your 95% of the population. So the first 95 states that the 95% of people should know their HIV status. So all of us would target that all of us know our own HIV status by conducting a test. And in 2023, it was achieved as 86%. So the lower range was 69, upper range was 98, and 86% know that their HIV status. But still, we are lagging behind because it has reached to 95. To reach the first 95, 90, 86 target, an additional 3.4 million people should know the HIV status of their own. So it's a huge number, 3.4 million know their HIV status. And coming to the second 90% of the people live that who know the HIV status, that this stands for the HIV positive people. So 90% of that HIV positive people should have received the antiretroviral therapy treatment. So in 2023, it acquires 77%, but still it is lagging behind the 90%. And to reach the second 95, 90, 86 target, around about 5.4 million people should have the target to know their status and access antiretroviral therapies. So this 5.4 million is also huge in number. Now coming to the viral load separations, so 86% should be there. So with the viral load, so the viral load has to be decrease. So in 2023, it has reached 72%. So still it is lagging to reach the 86%. So to reach this 86%, 5.6 million people with the HIV status report or the HIV positive should access the antiretroviral therapy. Then only it will reach the 95, 90, 86 target by the World Health Organization. So our main motto is to know the HIV status so that the HIV positive will get the antiretroviral therapy treatment properly, and ultimately it will reduce the viral load. So why reducing the viral load is so much important, we will discuss later on. So here, what we find is that there are two genotypes of HIV, that is HIV 1 genotypes and HIV 2 genotypes. Sorry for this. This one will be HIV 2 genotypes. Sorry for these mistakes. Now, the main difference between the HIV type 1 and type 2 was that that was the BPR and the BPX proteins which are present in that nucleotide sequences. Now, coming to there are three HIV type 1 genotypes. Then that is named as the M. M stands for the main, the most common one, and the O, which is just not the common one, outlier, and the new one, N. So the M, that is the most common one, comprised of a large number of subtypes, and that is around about 11 subtypes are there, A to K, and there are also recombinant forms. So there will be the mixing of these different variants forms of the main type of HIV-1. So there will be the diversity of the HIV virus. Now coming to the HIV two genotypes, there are still 8 subtypes and they are most prevalent in the West Africa, and here are 8 subtypes are there, A to H. Now, if we see the prevalence of this HIV type 1 and type 2, the red color signifies the C type, and the green color signifies the B type, and the blue color signifies the A type. So here we can see from the diagrammatic representations that the C type is most prevalent in the Indian subcontinent. So red color C type is more prevalent, but B type is also there and the A type is also there. And if we talk about the B type, they are mostly prevalent in the Europe, America, and the other Southeast Asian. And for the blue color, which is your A type, they are mainly prevalent in the West Africa. So now coming to our today's main topic, that is the lab diagnosis. So far there have been the advancement in the science and technology that the laboratory diagnosis has been creating for the specific and the non-specific tests. Non-specific tests are the indirect evidences related with the cell blood count or the T cell lymphocyte, T helper cell, CD4 plus cell counts are there. And here we are going for the specific tests, and these are the detections of the antibody test specific IgM or IgG to the HIV virus. So here the available tests in the market are the ELISA, enzyme immunoassay, Western blot, and the line immunoassay. Each has got a different principles, mechanisms, and the JMitra and Co. Private Limited has advanced in this ELISA. Now coming to the rapid antigens, what is the difference between this antibody test, other antibody test, and this rapid antigen or antibody test is that this rapid antigen antibody test will give you the results within 30 minutes. And however, this ELISA and then immunoassay, Western blot, and LIA, they require lots of durations of hours, and they require lots of resources, and they require the experience technicians or the healthcare setups to establish these resources. Now, there are another two types of these rapid antigen or antibody tests. Mostly the rapid antigen or antibody tests are conducted by using the blood from a venous. And the another one, which is now in creating a promising test, and that is the rapid antigen or antibody test by using the blood from a finger prick. So this rapid antigen or antibody test using the blood from a finger prick, here it makes everyone easy to have conduct these to know their status by alone, or and it is portable devices, and they can carry in a pocket also. And the more advanced technology, lots of resources are required, is your nucleic acid amplification test, in which PCR is also one of them. Now, what the JMitra and Co. Private Limited has come up with, that the another generation type of these rapid antigen and antibody tests, and this is known as the fourth generations. In these fourth generations, why it is known as fourth generations? Because they are enabled to identify the antigen as well as the antibody at a time. So this is the advancement of the science and technologies, and it is used up by the JMitra, and they are coming up with these diagnostics purposes. So if this diagnosis purposes is enhanced by this JMitra and Co Private Limited by emphasizing the use of the finger prick as a sampling devices. So not only with the blood vanners, they are coming up with the finger prick blood samples for this test device. So they are a combo pack of the antigens as well as the antibody. So it is just the upper one slot will be detecting the HIV antigens, and this when we inoculate or when we insert the samples either from the finger prick or from the venous blood, when we put a drop of that, then this pink color stands for the control. And by chance, if the person is suffering from the infections, there might be the color band stand up for the antigens as well as for the antibody. So this is the one of the promising and advanced rapid diagnostic test kit devices which are coming up by the JMitra and Co Private Limited. Not only for detecting the HIV status, it will help mostly in the prognostic treatment of the diagnostic purposes as well as the prognosis of these public health impact issues of this HIV. So the take-home messages to all I would like to give you to that HIV is not airborne and cannot be caught by the touching skin or sweat or saliva. You cannot get HIV by holding hands or sharing drinks. And please remember that the mosquitoes do not inject the other people's blood when they bite, so they can't spread HIV. A person has AIDS only in the final stages of HIV, so you must not be panicked. So there are lots of cases nowadays that the retroviral drugs has so much advanced that the HIV positive patients are still living beyond 15 years of ages of acquiring the HIV. So they are if they are maintaining a good health life, they can live on for a more number period of years. Previously, since due to not advance in so much of antiretroviral drugs, so there is high chances of acquiring these AIDS. So nowadays, we must not panic about that. If someone has got the HIV, then there will be the person will definitely suffer. They will not definitely suffer from the AIDS. Thank you for your kind attendance, and this is my if anyone has a doubt, you can clarify with me.
Thank you very much, Dr. Longjam. I think this was a very, very detailed study that you have conducted, and the kind of timeline that you took us through with the advent of HIV, with the advent of HIV, the understanding of HIV, and the fact that HIV is not equal to AIDS. The
Yes.
common misconceptions that you have helped to clear up today. Very detailed, and I think whatever little doubts that people would have had would have been very well clarified out here on today's forum. I want to share with you here that while we look at rapids and ELISAs as good testing mechanisms, we at JMitra have also launched the first made in India product, and we have got it cleared by CDSCO as well as by National Institute of Biologicals for HIV fourth generation testing on the chemiluminescence platform as well. This will not only speed up the HIV testing, but it will increase the sensitivity and the accuracy of the diagnosis. So while HIV remains a real world problem even today, I think the impact of HIV and its progression towards AIDS has been, controlled to a large extent due to a lot of research and treatments that
Exactly.
are now available. So we should not let down our guard. That's the message I want all our forum members to take away from here today. Do not let down your guard. Please be careful, especially when you're getting in terms of blood transfusions, or you're going in for, do not have unprotected copulatory measures. You should make sure you are protected, and it will keep you safe and live a happy and long life. Right. Thank you, Dr. Longjam, and we would love to partner with
Thank
you
you, sir.
again on other such subjects, which would be of mutual interest to all of us. Thank you, everybody, for attending it today. And we look forward to seeing you on the 28th, that is 2 days from now, on Saturday afternoon at 3 p.m. for our next webinar on serological techniques by Dr. Ramesh from Kerala. Thank you all. Have a good day. Jai Hind.